Today in this class we discussed about the next instrument, DNA synthesizer. This is the structure of DNA synthesizer. DNA synthesizer are machines used to custom build DNA molecules to contain a particular sequence of nucleotides. DNA synthesizer can create specific DNA molecules for use in the treatment of variety of diseases by replacing the faulty or damaged section of DNA with a repaired section. And DNA synthesizers use methods developed by scientists conducting the Human Genome Project. And the Human Genome Project provided a map of the entire human genome, enabling scientists to decipher specific sequences of nucleotides that are responsible for certain types of protein synthesis in the body. And the DNA synthesis is the process of copying a strand of DNA in a biological cell before the cell replicates or divides. Since the base pairs in the DNA always contain the same two complementary nucleotides, that is, adenine always bonds with thymine and cytosine always bonds with guanine. And DNA synthesizers can build DNA molecules using one strand of DNA as a blueprint and then use the DNA protein synthesis to repair any discrepancies in the strand. And DNA sequencer are DNA synthesizers that are used to analyze the sequence of nucleotides in the DNA sample. A DNA sequencer is often used to analyze DNA evidence from the crime scenes to provide a definitive DNA fingerprint of a potential suspect. And DNA sequencing is accomplished most often by the dideoxy or chain termination method. In this method, normal DNA protein synthesis is interrupted periodically by the introduction of a compound called deoxynucleotides triphosphate, which is substituted as the DNA chains is elongated by the addition of the normal nucleotides. These dideoxy compounds fluoresce under examination by a laser beam functioning like tags to mark out specific sections of the DNA molecules. Each section or DNA sequence can be separated and analyzed. And DNA synthesizers are available from several biological instrument manufacturers. DNA synthesizers may be programmable to handle batch analysis and are typically differentiated by cycle times and sample capacity and purity. And the next instrument is column chromatography. This is the structure of column chromatography. Column chromatography is one of the most useful method for separation of purification of both solids and liquids. This method is a solid liquid technique in which the stationary phase is solid and mobile phase is liquid. The principle of common chromatography is based on differential adsorption of substance by a adsorbent. The usual adsorbent employed in the column chromatography are silica, alumina, calcium carbonate, calcium phosphate, magnesia, starch. A selection of solvent is based on the nature of both the solvent and the adsorbent, and the rate at which the components of a mixture are separated depends on the activity of the adsorbent and polarity of the solvent. If the activity of the adsorbent is very high and polarity of the solvent is very low, then the separation is very slow but gives a good separation. On the other hand, if the activity of the adsorbent is low and polarity of the solvent is high, the separation is rapid but gives only a poor separation, that is, the components separated are not 100% pure. The adsorbent is made into slurry with a suitable liquid and placed in a cylindrical tube, that is plugged at the bottom by a piece of glass wool or porous disc. The mixture to be separated is dissolved in a suitable solvent and introduced at the top of the column and is allowed to pass through the column. As the mixture moves down through the column, the components are adsorbed at different regions depending on their ability for adsorption. The component is greater adsorption power will be adsorbed at the top and the other will be adsorbed at the bottom. The different components can be dissolved and collected separately by adding more solvent at the top of this process is known as elution. That is, the process is dissolved out of the components from the adsorbent is called elution and the solvent is called the eluent. The weakly adsorbed component will be eluted more rapidly than the other. The different fractions are collected separately. Distillation or evaporation of the solvent from the different fractions give the pure component. And the next instrument is ELISA. This is the structure of ELISA. Expansion is the enzyme linked immunosorbent assay, that is ELISA. ELISA is a common laboratory technique which is used to measure the concentration of an analyte in the solution, usually antibodies or antigen. And ELISA test. These are the variation of the ELISA test, but the most basic type consists of an antibody attached zero a solid surface. This antibody has affinity for the substance of interest. For example, human chorionic gonadotrophin, that is HCG, the commonly measured protein which indicates pregnancy. A mixture of purified HCG linked to an enzyme and the test sample, blood or urine sample, are added to the test system. If no HCG is present in the test sample, then only HCG with linked enzyme will bind the more HCG that is present in the test sample. The less enzyme linked HCG will bind. The substance on which the enzyme acts is then added and the amount is product is measured in some way, such as change in color of the solution. And procedure. Generally, ELISA is a 5-step procedure. First one, coat the micro titer plate wells with antigen, and second one, block all unbound site to prevent false positive results, and third one, add primary antibody to the wells, and fourth one, add secondary antibody conjugated to an enzyme, and fifth one, a reaction of the substrate with the enzyme to produce a color product, thus indicates a positive reaction. And last one, application and advantages of an ELISA. ELISA tests are widely utilized to detect substances that have antigenic properties and primarily proteins. The substances detected by ELISA test include hormones, bacterial antigens, and antibodies. And ELISA tests are generally accurate tests. They have the added advantages of not needing radioisotopes or a costly radiation counter. Compare with other method used to detect substance in the body, such as radioimmunoassay test. Thank you.